Picture this. It is 7:15 in the morning. You are standing in your kitchen, still half asleep, reaching for the coffee maker on autopilot. You've done this thousands of times. You think of it as a habit, a ritual, a necessity. What you've never thought about is what happens in the next 30 to 90 minutes, inside an organ you cannot see or feel, sitting quietly under your right ribcage, processing everything that arrives from your digestive system before it reaches the rest of your body, your liver. Because every single time you drink coffee, not occasionally, not in excess, but every ordinary morning cup, something specific and measurable happens inside your liver. Something that researchers have spent decades trying to fully understand because the findings keep defying what medicine expected to find. The story of coffee and your liver is one of the most surprising chapters in modern nutritional biology. And it begins not with caffeine, which most people assume is the active player, but with a group of compounds so obscure that most people have never heard their names. Let's go inside. Your liver is the most metabolically complex organ in your body. It performs over 500 distinct biochemical functions simultaneously, 24 hours a day, without pause. It processes every nutrient that enters your digestive system before releasing it into general circulation. It manufactures bile for fat digestion, produces the clotting factors your blood needs, synthesizes cholesterol and then regulates it, converts ammonia from protein metabolism into urea for excretion, stores glycogen for blood glucose regulation, and runs the primary detoxification pathways that process medications, alcohol, environmental toxins, and the metabolic waste products your own cells generate. It is, in every meaningful sense, the chemical processing plant that keeps your entire internal environment stable. And it is an organ under extraordinary stress in the modern world. Non-alcoholic fatty liver disease now affects approximately 25% of the global adult population, 1 in 4 people, driven by the combination of excess fructose, chronic inflammation, insulin resistance, and sedentary behavior that characterizes modern life. Liver cirrhosis, the endpoint of chronic liver damage regardless of cause, kills over 1 million people annually worldwide. Against this backdrop, the discovery that an ordinary daily beverage produces measurable protective effects in liver tissue is not a minor finding. It is one of the most clinically significant observations in nutritional epidemiology of the last 2 decades. The first thing coffee does inside your liver arrives within minutes of absorption. Coffee contains over 1,000 distinct bioactive compounds, but the ones most relevant to liver biology are the chlorogenic acids, a family of polyphenols that are among the most abundant antioxidant compounds in the human diet for coffee drinkers. Chlorogenic acids are absorbed in the small intestine and travel directly to the liver through the portal vein, the same route that all nutrients from digestion take. Inside the liver, chlorogenic acids activate a cellular signaling pathway centered on a protein called NRF2, nuclear factor erythroid 2 related factor 2. NRF2 is your liver cell's master antioxidant switch. When activated, it triggers the production of a cascade of endogenous antioxidant enzymes, glutathione, superoxide dismutase, hemoxygenase, that neutralize the reactive oxygen species produced by your liver's own metabolic activity. Every time your liver processes nutrients, detoxifies compounds, and runs its 500 biochemical functions, it generates oxidative stress as a byproduct. Chlorogenic acids from coffee activate the precise cellular machinery that cleans up this oxidative stress before it accumulates and damages hepatocytes, your liver cells. This is not supplemental antioxidant activity layered on top of your liver's normal function. This is the activation of your liver's own internal antioxidant defense systems by a compound that arrives from your morning cup. The effect is measurable in blood markers within hours of consumption and cumulative over days and weeks of regular intake. The second effect operates through a completely different pathway and is arguably the most clinically significant. Coffee consumption, specifically the combination of chlorogenic acids, caffeistol, kahweol, and other diterpenes present in coffee has been shown in multiple large-scale epidemiological studies and mechanistic laboratory research to reduce liver fibrosis. Fibrosis is the process by which healthy liver tissue is replaced by scar tissue, fibrous collagen deposits that permanently reduce the liver's functional capacity. It is the intermediate stage between liver inflammation and cirrhosis. Once significant fibrosis is established, it is largely irreversible through dietary means. Preventing it is therefore of far greater clinical importance than treating it. The mechanism by which coffee reduces fibrosis involves the activation of hepatic stellate cells, the cells responsible for producing the collagen that forms fibrotic tissue. Under normal conditions, hepatic stellate cells remain dormant. When the liver is chronically inflamed, damaged by alcohol, fat accumulation, or toxins, stellate cells activate and begin producing collagen fibers. Coffee compounds, particularly cafestol and cawiol, suppress the activation of hepatic stellate cells through inhibition of the TGF-beta signaling pathway, the primary molecular signal that tells stellate cells to switch from dormant to fibrosis-producing. By blocking this signal, coffee effectively reduces the liver's tendency to replace damaged tissue with permanent scar tissue. A 2017 meta-analysis covering over 800,000 participants across multiple countries found that every additional cup of coffee per day was associated with a 7% reduction in the risk of liver cirrhosis. 2 cups per day was associated with a 44% reduction in cirrhosis risk compared to non-drinkers. These are not marginal statistics. These are among the most consistent and robust findings in nutritional hepatology. The third effect involves fat accumulation in liver tissue, the defining feature of non-alcoholic fatty liver disease. When fat accumulates inside hepatocytes, it impairs their function, generates oxidative stress, triggers inflammation, and initiates the fibrosis cascade. Coffee consumption reduces hepatic fat accumulation through several converging mechanisms. Chlorogenic acids reduce de novo lipogenesis, the synthesis of new fat from carbohydrates, in liver cells by inhibiting key enzymes in the lipid synthesis pathway. They also activate AMP-activated protein kinase, or AMPK, the cellular energy sensor often called the metabolic master switch, which promotes fat oxidation and suppresses fat storage in liver tissue. Caffeine independently activates AMPK through a separate pathway, meaning that both caffeinated and decaffeinated coffee produce some benefit through chlorogenic acid pathways, but caffeinated coffee produces a stronger combined effect through dual AMPK activation. A 2012 study published in the journal Hepatology found that coffee consumption was associated with significantly lower rates of non-alcoholic steatohepatitis, the inflammatory form of fatty liver disease, even after controlling for other dietary variables, exercise, and metabolic risk factors. The protective effect was dose-dependent, stronger with higher consumption up to approximately 4 cups per day, and independent of whether participants had pre-existing metabolic conditions. The fourth effect operates through your liver's relationship with inflammation at the systemic level, where liver produces the majority of your body's acute phase proteins, the inflammatory signaling molecules that respond to tissue damage, infection, and metabolic stress throughout your body. Chronic, low-grade systemic inflammation, the kind that underlies metabolic syndrome, cardiovascular disease, and accelerated aging, is partly driven by the liver's continuous production of pro-inflammatory cytokines in response to the metabolic inputs it receives. Coffee consumption reduces hepatic production of several key pro-inflammatory markers, including C-reactive protein, interleukin-6, and tumor necrosis factor alpha. A large analysis of data from the National Health and Nutrition Examination Survey found that coffee drinkers had significantly lower serum CRP levels than non-drinkers, independent of other lifestyle factors. This anti-inflammatory effect is not incidental to coffee's liver biology. It is a direct consequence of the reduction in oxidative stress stellate cell activation, and lipid accumulation described above. When the liver is less inflamed and functioning more efficiently, its production of systemic inflammatory signals decreases accordingly. The fifth effect is the one that surprised researchers most and generated the most clinical interest. Coffee consumption is associated with a significantly reduced risk of hepatocellular carcinoma, primary liver cancer. Liver cancer is the sixth most common cancer globally and the third leading cause of cancer death. Its primary risk factors are chronic hepatitis B and C infection, alcohol-related liver disease, and non-alcoholic fatty liver disease progressing to cirrhosis. A 2013 meta-analysis examining data from over 2 million participants across 22 studies found that 2 cups of coffee per day was associated with a 40% reduction in hepatocellular carcinoma risk compared to non-consumption. The mechanism is multifactorial. Reduced fibrosis progression means fewer cirrhotic livers, which is the primary risk factor for liver cancer. Reduced oxidative stress means less DNA damage in hepatocytes. Reduced chronic inflammation means less of the pro-carcinogenic cellular environment that promotes malignant transformation. The NRF2 activation pathway itself has anti-tumor effects, promoting the elimination of pre-malignant cells through apoptosis. The protective effect against liver cancer is one of the most consistently replicated findings in coffee research, and one of the most clinically meaningful given the severity of the disease. Here is what all of this means in practical terms. The research does not suggest that coffee is a treatment for liver disease. If you have existing liver disease, significant fibrosis, or cirrhosis, coffee is not a substitute for medical management. What the research suggests, consistently, across enormous data sets, through multiple biological mechanisms, is that regular, moderate coffee consumption in healthy adults produces a measurable protective effect on liver tissue that operates through at least 5 distinct biological pathways simultaneously. The optimal range in the research is between 2 and 4 cups per day. Beyond 4 cups, the incremental benefits plateau, and the side effects of excess caffeine, elevated cortisol, disrupted sleep, increased heart rate, begin to offset the liver benefits. Preparation method matters less than most people assume. Filtered coffee, espresso, French press, cold brew, and instant coffee all deliver chlorogenic acids in meaningful amounts. The primary variable is consumption volume and consistency. Decaffeinated coffee retains most of the chlorogenic acids and produces similar but somewhat attenuated liver benefits through caffeine-independent pathways. Adding large amounts of sugar or industrial seed oils to coffee, as in many commercial coffee drinks, partially offsets the benefits by introducing the exact metabolic stressors that coffee's liver-protective compounds are counteracting. Your morning coffee is not just a habit. It is not just caffeine delivering wakefulness through adenosine receptor blockade. For your liver, it is a daily delivery of bioactive compounds that activate antioxidant defense systems, suppress fibrosis signaling, reduce hepatic fat accumulation, dampen systemic inflammation, and reduce the risk of one of the world's most lethal cancers. None of this means you need to drink coffee if you do not enjoy it. But if you do drink it, understanding what it is actually doing inside your liver every single morning is the kind of biological knowledge that changes how you relate to something you have done on autopilot for years. Your liver has been benefiting from something you thought was just a ritual. If this changed how you think about your morning cup, subscribe to this channel. Every video here goes inside the actual mechanisms, so you understand not just what to do, but exactly why it works. Like this video and share it with someone who feels guilty about their daily coffee. Show them the biology has been on their side the entire time.